Hello all, welcome back to another video tutorial. Today's topic is the benefits and risks of PEGylation in nanomedicine. Polyethylene glycol is a popular coating agent in nanomedicine, valued for its ability to improve the performance and safety of nanoparticle-based drug delivery systems. However, recent studies have raised concerns about its potential immunogenicity. In this section we will discuss about the few scientific comments related to benefits and risks of PEGylation in nanomedicine. A major challenge in making nanomedicines work in real patients is controlling how the body sees them. As soon as nanoparticles enter the body, they get covered by layers of the body's own proteins. This is called the protein corona. The protein corona affects whether the nanoparticles can avoid being quickly removed by the immune system, as well as how well they can reach and enter their target cells. A common way to control the protein corona is to coat nanoparticles with PEG, a substance that helps them hide from the immune system and stay in the body longer. PEG is safe for humans and is used in many approved medicines by the U.S. Food and Drug Administration, and favorable physiochemical properties have made it the polymer of choice for nanomedicine surface modification. However, PEG also has some downsides. It can make it harder for nanoparticles to find and stick to their target, and in some cases, it can cause immune reactions. This trade-off between the stealth properties given by PEG and the targeting efficiency provided by antibodies is a key argument in the scientific community. To get the best results, scientists try to find the right balance by adjusting things like the ratio of PEG to antibodies, the length and weight of the PEG chains, and how the antibodies are attached. Another approach is to design a special artificial protein corona to better understand how natural protein coronas form, which could help direct nanoparticles to specific organs. Another major argument in the scientific community concerns the immunogenicity of PEG. Anti-PEG antibodies were first discovered in animals exposed to PEGylated proteins in the 1980s. Later studies linked these antibodies to reduced stealth properties and lower effectiveness of PEGylated nanomedicines, as the immune system could clear them from the body more quickly. This issue has been recognized for years but became especially important during the COVID-19 pandemic, when some people experienced hypersensitivity reactions to PEGylated lipid nanoparticles used in vaccines. While researchers are exploring polymer alternatives to PEG, it is likely that other polymers could also cause immune responses over time, leading to similar problems. One possible clinical solution is to alternate between different stealth polymers to help prevent the immune system from developing strong reactions to any single one. Another important issue is that alternatives to PEG might also trigger immune responses, and there are still big gaps in our understanding that need to be addressed before switching to new materials. First, there is no standard way to test for antibodies that bind to these polymers. Sometimes, researchers use the term immunogenicity incorrectly, referring to cytokine production, a sign of general immune activation, but this does not always mean that the body is making specific antibodies against the polymer. It's important to clearly distinguish between a product that causes antibody production, immunogenic, and one that just stimulates cytokine release, immunostimulant. Second, we need a better understanding of how PEG and similar polymers actually cause the immune system to react. Not all antibodies are the same. Different types can have different effects on the body. Also, whether PEG is free, attached to a protein, or attached to a nanoparticle can change how the immune system responds. Experts also note that there are already regulatory guidelines for testing immune reactions to biologic drugs, including PEGylated proteins. Even though these rules were made for small drugs, the nanomedicine field should use similar methods to study and manage immune risks from PEG and other coatings. Every PEGylated product, and every nanomedicine with a different surface coating, should be evaluated individually, looking at both antibody-related and non-antibody-related risks in its specific context. By using the right tools and deepening our understanding of how these polymers interact with the immune system, scientists can more accurately assess risks and design safer, more effective nanomedicines. This will help ensure that the benefits of PEG and its alternatives outweigh the risks. Thank you for watching. Stay tuned for more updates.